Germ theory introduced the idea that tiny microorganisms called germs are responsible for many diseases. These germs included bacteria, viruses, and fungi, which can spread from person to person through the air, water, food, or physical contact. Before germ theory, many people had believed diseases were caused by bad air, curses, or an imbalance in the body. Doctors often treated patients without understanding what really made them sick. The reason these theories survived for so long is that scientists and physicians didn't have anything better to replace them with. But this all began to change in the 1800s. A huge turning point for medicine was cell theory in 1837. This determined that every living thing was made up of cells. Now, cells were not a new discovery. Microscopes became very sophisticated very quickly, and in 1667, Robert Hooke published his famous Micrographica. Within this book was the first use of the term cells. His observation of cork gave us the first look at plant cells. Dutch draper Antony van Leeuwenhoek was the first person to use a microscope to observe and describe bacteria, yeast, blood cells, and microorganisms, which he famously termed animalcules. This was around 1675, so centuries before germ theory. In 1840, anatomist Jacob Henle argued that disease was not caused directly by bad air, but by microscopic living organisms carried through the air. Though he believed proving this would be impossible, scientists like Louis Pasteur and Robert Koch played a major role in proving his theory during the 19th century. Louis Pasteur was a French scientist whose discoveries transformed medicine and microbiology. He proved that microorganisms cause fermentation and disease. He also disproved the idea of spontaneous generation, which is a theory that believed living creatures could regularly arise from non-living matter. His major breakthrough was showing that specific disease is associated with a specific bacteria, although cause and effect relationship between specific germs still had not been determined. And then there was Pasteur's German rival, Robert Koch. Koch was a student of Jacob Henle, and it was Koch whose research turned into the experiment that Henle deemed impossible. Henle believed that you could only extract bacteria from dead animals, but whilst researching an outbreak of anthrax in livestock, Koch successfully extracted tissue while the animal was still alive. Within living tissue, the bacteria could keep multiplying, resulting in an endless supply of uncontaminated bacteria. Take this bacteria, culture it, which means let it grow and reproduce in a controlled environment, inject the bacteria into a new animal subject, and see what happens. This was the first time someone had isolated a microorganism and caused disease with it. Whilst Koch was publishing his work on anthrax, Pasteur started working on a vaccine for it. This was not done as a collaborative effort. The two men despised each other, partly because of the tensions between France and Germany at the time and their differential approaches to microbiology. It also does look like Pasteur plagiarized some of Koch's work, so despite their beef, the two men discovered ways to identify disease on a cellular level, identify the causative agents of the disease, culture bacteria to enable it to be studied in a controlled manner, and develop vaccines and cures for diseases like tuberculosis, plague, dysentery, and tetanus. Germ theory completely transformed healthcare. Hospitals became safer, surgeries became more successful, and millions of lives were saved.